All right, good morning, everyone. So today I'm going to talk about the control of shoot branching plasticity by different sugar signaling pathways. So as you can see here, there is a defined architecture on plants. And here, I used Arabidopsis as an example because it's also the model we use in the lab. But this plasticity, this architecture depends on different factors. So, for example, here, the one I've been showing has been grown in what we call the regular lab condition. So, long day and 150 microeinstein of light. But now, if I grow the same genotype under low light, I've got a quite different shoot branching architecture. Or if I mutate MAX4, which is a gene controlling strigolactone synthesis, we've got a very strong shoot branching architecture. So which shows that shoot branching is regulated by environmental and genetic factors. So in plants, shoot branching is controlled by axillary bud outgrowth. So little buds which are at the axil of each leaf at each node. And each axillary bud is made of a meristem with primordia and internodes which are not elongated yet. So it's a compact structure which is just ready to grow when the bud is released from dormancy. And so one of the mechanism or phenomenon which controls shoot branching is apical dominance, which is the inhibitory effect of the growing shoot tip over the axillary buds down the stem. And therefore, it's also controlled by, it's partly controlled by a flux of auxin, which is produced by the growing shoot tip and which inhibits axillary bud outgrowth. And therefore, when you decapitate the shoot tip, the buds grow out into branches. So locally, what happens is that auxin does not enter the bud and instead regulates cytokinin and strigolactone synthesis, which regulate bud outgrowth. So the problem with auxin being the only signal to control apical dominance is that there's a decorrelation between auxin dynamics and bud outgrowth. And for example, Christine's group has shown a few years ago, and we repeated that recently, that when you decapitate the plant, the auxin depletion in the stem does not match the dynamics of bud outgrowth. So there's another signal which is earlier than auxin, which triggers the bud to grow out. And so in 2014, Christine's work has shown that sugar availability was a major signal to trigger bud outgrowth in response to decapitation. And so the role of sugar availability in shoot branching is not limited to apical dominance. And in conditions where the plant is intact and photosynthesis fluctuates due to different environmental reasons, you also have a control of shoot architecture due to modulation of sugar availability to the buds. So in eukaryotes, and actually in all the domain of lives, sugars act like signals, like hormones do. And my research goal in Christine's group was to understand the sugar signaling pathways which control shoot architecture. So one way we have to do this is to use non-metabolizable sugars. So because the problem with feeding sugars is that you've got the signaling component, but also the metabolic component, unlike hormones, which only have a signaling component. But if we use sugars which are analogs of certain sugars and which are not metallizable, we can trigger the signal without triggering the metabolism. And so, for example, in this work that I did in Rose before arriving to Australia, we used single internode with one single node, and we fed different sucrose analogs. And so we did that in different species, Rose P and Arabidopsis. And so what I ask you to see here is just that the control is at the bottom, so the buds are not growing out, and that sucrose and its analogs, which are not going through metabolism, can trigger bud outgrowth, showing that sucrose is involved in a signaling manner during shoot branching. So there's an obvious sucrose signaling pathway which is involved or which exists in plant, which is mediated by Trehalose 6-phosphate, which acts like a hormone. So I'm not going to talk about that today because Francisca already gave several talks about T6P and shoot branching, and her work has shown that T6P can trigger shoot branching in P and Arabidopsis. So I'm going to spend a bit more time on hexokinase. So hexokinase is the first enzyme of the glycolysis. and also has a signaling function, notably due to the fact that it can go to the nucleus and regulate the transcription of some target genes. And so here I use the same approach that I did with sucrose. I use glucose analogs that I fed to single P nodes. And first I fed glucose, so which is the endogenous substrate of hexokinase. And as you can see here, it can trigger axillary bud outgrowth. Now, when I feed mannose, which is a glucose analog, which is also phosphorylated by hexokinase, but is not further metabolized, we could see that we can also trigger bud outgrowth. However, when I used a 3-O-methyl glucose, which is also a glucose analog, it enters the cell, but is not phosphorylated by hexokinase and is not metabolized. And this time, the bud outgrowth was very small compared to the two other sugars, suggesting that hexokinase is involved in a signaling manner independently of its catalytic activity. And so we tested this also in our Arabidopsis at the genetic level. So I used the gene2 mutant, which is a glucose insensitive mutant, which doesn't have hexokinase activity or signaling. And when I scored the number of rosette branches which emerged from the rosette after bolting, I could see that this mutant produces a lot less branches than the wild type, Landsberg. And to show that hexokinase was involved in a signaling manner, here we complemented gene2 with a hexokinase which has a point mutation, which makes that it cannot phosphorylate glucose, but it still retains its signaling activity. And when we complemented gene2 with this hexokinase, we could see that most of the branching phenotype was restored, showing at the genetic level that hexokinase was involved in a signaling manner in this process. So as I mentioned before, auxin regulates bud outgrowth through controlling cytokinin and strigolactone. And my goal was also to understand how sugars control the cytokinin and strigolactone pathways during bud outgrowth. So what we've shown in ROSE in 2015 was that sucrose and also its analogs can trigger the accumulation of cytokinin in the stem and the buds compared to the mannitol used as a control. And so I decided to measure cytokinin in a gene2 mutant. And we could also observe that the gene2 mutant has less cytokinin, notably the zeatin form of cytokinin, which accumulate less in this mutant compared to the wild type. So we tried to test if part of the decreased branching of gene2 was due to a lack of cytokinin. So we decided to treat axillary buds. So in Arabidopsis, they are very, very tiny at the axil of the leaves in the rosette. And so as you can see here, the buds of gene2 grow a lot less than the wild type. And now when I treat these buds with BAP, which is a cytokinin, I could restore parts of the decreased bud outgrowth of gene2, suggesting that part of the gene2 debranched phenotype is due to a lack of cytokinins. So now to our model, we can start to add that sucrose act at least partly through cytokinin, and that part of this effect is mediated by hexokinase 1. And we also worked on testing the impact with a collaborator, Danny Eshel, and a PhD student, BJ Salam. So we tested the impact of cytokinin on sucrose sink strength in the bud. So sink strength can be regulated by invertase activity. And so the works, I'm not going to show the results, but our work has shown that cytokinins were triggering invertase activity to get sucrose to enter the bud and to promote this effect. So it seems to be that there is a positive feedback loop between sucrose and cytokinin during the promotion of bud outgrowth. So now, what about the interaction between sugars and strigolactones? So we use the same system as before to test the impact of strigolactone and sucrose on bud outgrowth. So we used P single nodes. So we treated the nodes with GR24, which is a form of strigolactone. And here we first fed them with quite low concentration of sucrose. And as you can see, GR24 has a strong inhibitory effect on bud outgrowth. But now, if I repeat the same experiment, having a huge amount of sucrose in the growth medium, you can see that GR24 does not inhibit anymore bud outgrowth. We could repeat that also in rose and in rice. And this is a collaboration that we did with Triangli and Suyash Patil. And so here we worked in rice, and we first repeated the same experiment, but in hydroponics. So we worked in entire plants, and we scored the number of tillers which emerge from the main tiller. And so we could see that GR24 has an inhibitory effect on tillering under low sucrose concentration. But suddenly, when you have high sucrose concentration, GR24 has a very small effect on tillering. So we wanted to understand how sucrose could alleviate the effect of strigolactone on shoot branching and tillering. So tillering is triggered by a protein called D53. And D53 is targeted for degradation by strigolactones, notably due to the fact that strigolactone forms a complex with its receptor, D14, and an F box called D3, also known as MAX2 in Arabidopsis. So we first looked at the response of D53 to sucrose. Using Western blot, we could see in rice kalae that D53 strongly accumulates in response to sucrose. And then we wanted to test the antagonism with strigolactones. So we used another assay, which is agro-transfected tobacco leaves. So here we used the luciferase D53. And when you transfect the leaf with this construct plus or minus strigolactone, so GR24, you can see that there is less accumulation of the protein, suggesting that strigolactone lead to the degradation of D53 in this assay. But this is in absence of sucrose. Now, if I repeat the same thing with 4% sucrose, you can see that the effect of GR24 on D53 has almost totally vanished, suggesting that, again, sucrose can alleviate the effect of strigolactone on D53. So now we want to understand how sucrose does this. So, sorry, I went a bit further. So we measured the expression of D3, so MAX2, which is the F box upstream of D53, which is responsible of D53 degradation. And we could see that feeding sucrose to different parts of the plant could inhibit the expression of D53, which is what we observed a few years ago in rosebuds in response to a range of sucrose. So we decided to repeat the experiment that we did before, but this time with co-transfecting D3, an overexpressor of D3. And contrary to what we observed without co-transfecting, this time GR24 retained its inhibitory effect on D53, suggesting that sucrose cannot anymore promote D53 if MAX2/D3 is overexpressed. So now we wanted to test this at the phenotypic level. So if you remember in wild type, when we had 4% sucrose, GR24 could not inhibit any more tillering in rice. But now if we repeat the same experiment by using a line which overexpresses D3, MAX2, this time GR24 retains its inhibitory effect on tillering, and sucrose cannot alleviate anymore this inhibitory effect, suggesting that MAX2 D3 is an important node in the antagonism between sucrose and strigolactones. So now we can place on our model that sucrose inhibit the strigolactone signal, notably through MAX2. And so now what we try to understand is what is the signaling pathway which enables sucrose to inhibit MAX2. So we looked at MAX2 promoter, and we could find a cluster of G boxes 4 kb upstream of MAX2. So we were interested in this cis element because it's known to be bound by BZP11, which is a transcription factor known to be inhibited by sugars through multiple ways. So we looked in DAPSIC data to see if BZP11 was binding on this locus, and we could see that indeed BZP11 seems to bind to this locus. And so during Alicia Helen's PhD project, we tested this further and tested the expression of MAX2 in response to BZP11 induction. And she could see that indeed, when you overexpress BZP11, MAX2 expression goes to the roof. So I'm not going to give more detail about this because we are still working on this. We think this interaction between sucrose and MAX2 through BZP11 is only a part of the mechanism, and we think there's way more to it. And actually, Isabella Amor, who's probably here today, is working on it at the moment, and we think that other metabolites, such as citrate, are involved in this regulation. However, through this approach, we've been able to highlight that BZP11 was involved in shoot branching. And so this time, we collaborated with Christoph Weister and Philipp Kreis in Wolfsburg University to test the involvement of BZP11 in shoot branching in Arabidopsis. So one of the first experiments we did was to decapitate Arabidopsis and to measure BZP11 protein levels in the core of the rosette, which is enriched in axillary buds. And so what we could see in different, so here you've got 5 replicates for each condition. And what we could see is that the protein level of BZP11 is strongly inhibited in decapitated plants compared to intact plants, suggesting again that BZP11 may be involved in shoot branching. So to test this, we generated a triple BZP11 mutant. So BZP11 mutant has 2 very close homologs, BZP2 and BZP44. So we used a triple knockout. And as you can see on this picture, the plants, compared to the wild type, the plant is quite similar, except that it flowers slightly later, but also produces more rosette branches compared to the wild type, further supporting that BZP11 is involved in shoot branching. So now to test further if BZP11, so this work is at the moment on bioarchive, is on the review for a journal. And to further test the impact of BZP11 on bud outgrowth this time, we used a choline assay. So it's the same assay that I explained before, where you take just one single node, except here we take the choline nodes growing on the rosette branches, and we put them in vitro. So here we used a BZP11, which can be induced to go to the nucleus upon dexamethasone treatment. So the protein is always expressed but stays in the cytoplasm until you put dexamethasone, which triggers BZP11 to go to the nucleus to have its function of transcription factor. And so when we fed the bud with dexamethasone, a range of dexamethasone, so it means the more dexamethasone you have, the more BZP11 activity you have. And we could see that bud outgrowth was repressed, suggesting that BZP11 is involved in bud outgrowth and shoot branching. So in this publication, we also highlighted the interaction between BZP11 and other pathways that we've shown to be involved in shoot branching, notably T6P. And also we highlighted that SNRK1, so it's a strong, it's a central kinase regulating or sensing sugar starvation and controlling development. And we could see that there was an interaction between these 3 signals to control bud outgrowth. I'm not going to show the results. And if you want to see more, it's on bioarchive at the moment. But all of this helps me to draw a model through which sugars act through hormone, through different sugar signaling pathways to fine-tune shoot branching. So obviously, this is a way for which the environment, through modulating photosynthesis, is going to modulate shoot branching due to the differential accumulation of sugars in the plant. But this is not, sugars are not the only nutrient and metabolite to control shoot architecture for this way. And we just highlighted in a review that the strigolactone pathway, for example, was a very strong integrator of different metabolic and nutritional signals. And for example, strigolactone signaling here at the bottom tends to be more controlled by metabolite like sugars or citrate, while nutrient like mineral nutrients like phosphate, nitrate, or sulfur tend to regulate strigolactone synthesis, showing that the whole pathway is controlled by metabolic and mineral cues to control shoot architecture. So I would like to thank my group, the Beveridge group, for all the help over the 9 years of my postdoc so far, and also all the collaborators who contributed to this. And this talk is also a bit of a goodbye for me, because I'm going to, in 2 months, I'm going to move to France to start a new position and work on nitrate and sugar in the control of plant development.